Hello, I'm your host Mike Ward and welcome to BioBiz Buzz, the podcast that tracks the trends in the life sciences and talks to the innovators driving those changes. What if the key to treating some of the most stubborn chronic diseases wasn't actually a new small molecule or biologic, but a precisely designed team of bacteria working in concert, developed and manufactured as one? Let me set the scene. The human gut is the home to trillions of bacteria, a community so complex and so influential that scientists are now calling it a virtual organ. It regulates immunity, metabolism, inflammation, even mood. When it tips out of balance, a state called dysbiosis, the consequences can be severe: chronic inflammation, autoimmune flares, and a growing list of diseases we're only beginning to trace back to the gut. Medicine has known this for decades, and for decades struggled to do anything useful with it. Fecal microbiome transplants were the first attempt: stool from a healthy donor, transferred to a sick patient. Sometimes it worked, but FMT is variable, hard to standardize, and carries safety concerns that make regulators and patients equally uneasy. Single-strain live biotherapeutics came next. One carefully chosen bacterium in a capsule, elegant, but a single actor rarely has the complexity to shift a deeply dysbiotic ecosystem back towards health. So the field landed on an obvious but devilishly difficult idea: design a team of bacteria, a consortium, selected to work together to target the specific mechanisms driving a specific disease. Rationally designed, optimized for synergy, manufactured to pharmaceutical standards, delivered as a single oral dose. Obvious in theory, in practice one of the hardest problems in drug development. But that's the founding idea behind MRM Health, a clinical stage biotech out of Ghent in Belgium. In this episode of BioBiz Buzz, I'm joined by Dr. Sam Possemiers, a scientist entrepreneur who has spent more than 20 years decoding the gut microbiome. He founded MRM Health to apply rational consortia design, proprietary manufacturing, and rigorous clinical development to the microbiome. Sam, welcome to BioBiz Buzz.
Hey Mike, thanks for having me.
So, Sam, you spent more than 20 years working on the gut microbiome. How would you explain its role in disease, to someone who still thinks of bacteria maybe as the enemy?
To kick off, maybe I could start with the fact that deep understanding of the role of microbiome in disease is actually still very recent science. It's literally only about 20 or even 15 years ago, which was more or less the time that I started working on microbiome. At that moment, pharma mostly focused on eradication of bacteria, and you could almost say the only good bacterium was a dead bacterium at that moment. But that has rapidly changed since then. There has been a rapid increase in the understanding that the balance between all those different bacteria that we have in our gut, that's not only key to maintain health, but also if there's a disturbance in that balance, which we call dysbiosis, that has a key role in a broad range of disease. So you could say that in the last 10, 15 years, there's a reframing of the role of those bacteria. And there are, of course, a number of highly pathogenic bacteria, but actually the vast majority is not doing harm to our body, but is actually an essential part. And you could say they are allies of our body. And you could compare the gut microbiome like a factory workforce, and they are producing a whole range of products such as short-chain fatty acids, which keep the gut lining in a healthy state, which train the immune system to distinguish friends from foe, keep harmful species in check through competition, and so on and so on. So that workforce acts as what we call an ecosystem, a team of bacteria, all interacting with each other and all working towards supporting the health of their host, because ultimately also for those bacteria, a healthy host is in benefit for that ecosystem. However, when that workforce is then disrupted through antibiotics, to diet, to stress, to disease, you name it, that factory breaks down, the teamwork is lost, and those key processes which are normally regulating our body are becoming disturbed. And as a consequence, we see that balanced interaction is lost, inflammation rises, the gut barrier weakens, the immune system starts misfiring, and we now know that there is a very direct link between disturbance of that microbiome and a range of diseases, starting in the gut with IBD, of course, but going all the way even up to neurological conditions, metabolic diseases, and beyond.
So the microbiome, it's not just along for the ride, it's an active driver of health and disease. So that brings us to the therapeutic angle. What exactly is a live biotherapeutic product, and how is it different from the sort of the probiotics that someone might pick up at the health food shop?
I think I would say that I mentioned already the breakthrough on the understanding of the role of the bacteria on disease, that the major breakthrough in the pharmaceutical context came that microbiome is not only involved in disease, but that you can actually use the good fraction of the microbiome to treat disease. And hence, a live biotherapeutic product is actually a medicine based on live bacteria. Typically, you derive them from the healthy individual, and then you bring one or more core commensal bacteria together. And those core commensals, those are, you could say, those high performance bacteria that you normally have in your guts and that play that central regulating role in the body. You bring them together in product and you develop it as a real drug. So you design the product, you test it, you manufacture it under GMP conditions, you validate it extensively in clinical trials, and ultimately you make it subject to the full regulatory scrutiny from FDA, from EMA, and so on. So an LBP is really a drug. Probiotic, in contrast, is a food supplement. So it's also developed in a different way. A probiotic does not need to demonstrate clinical efficacy against the disease. It's not held to manufacturing standards. Typically for pharma, you could rather say that the strains are typically chosen to maintain general gut health or general immune health, rather than that they are capable of treating the disease with a defined mode of action.
Yeah, I guess that's a sort of a distinction that a lot of people miss. Now, earlier sort of approaches, whether it's FMT or single strain therapies, and I'm just wondering, so why have those struggled to become mainstream medicines?
As I mentioned before, the microbiome field as a field, it's still a very novel field, and therefore the modality is also still very novel. And like any novel modality, it's associated with a steep learning curve. What works best? How do you need to design it? How can you dose it, etc.? And so it started with FMTs, and FMTs, could say it's fundamentally a black box. Right? You transplant an entire ecosystem with hundreds, if not thousands, of different species. You bring them into the patient without knowing exactly which ones are now the one that will do the job and which ones are the one that they're therapeutic, which ones are just passengers or may even be harmful. They may even be safety risks, for instance, like antibiotic resistance transfer, etc. But of course, also, you can't produce it, you can't manufacture it at a large scale. So it's impossible to give this to patients in large indications where you need to treat thousands or even millions of patients and where you need to keep dosing those products in a chronic fashion. And in a complex chronic disease like, for instance, UC, which is multifactorial, there are different things coming into play. You need to restore barrier function, you need to modulate immunity, you need to produce anti-inflammatory metabolites, you need to outcompete pathogens. So you need to do a whole lot of things in parallel, which is very tricky and difficult to do with one specific bacteria. So there were these initial approaches, and what we see now is that the field needed like a third path, which positions itself actually in between both.
Yeah, I suppose that third path is where MRM Health actually comes in. Your Coral platform, it sits at the heart of everything that you're doing. So could you actually walk us through what Coral actually does that wasn't possible before?
So first of all, Coral is an end-to-end discovery and development platform. So it's not a single machine. It's rather, you could see it as a toolbox of know-how, of different technologies which are integrated to allow the full design, development, and manufacturing of those consortia therapeutics, which are optimized to treat disease. Now, if you break it down, you could split it in 4 main stages, which are all integrated. First one is, of course, about the building blocks. What will you make the product with? So in our case, that means patient-driven strain selection. So you need to identify those strains which really make a difference in treating the disease. This is done largely based on bioinformatics analysis of patient microbiome data, in which we can identify strains which are depleted in the patient, and more importantly, which functions are actually missing and which are linked to the key disease mechanisms. So it's not about randomly picking strains from catalog. It's really function-based, careful selection of key strains based on those disease mechanisms. So that's the building block. Second step is then when you have those multiple building blocks, you start building your team and you start building that combination of strains that ultimately form your consortium. In our case, it's about 5 to 30 strains that we then bring together and that we really, you could say, literally develop as a team. It's you could say a team building at micro level to boost the synergy between the strains, to make a very robust product, and to also make sure that every strain in a consortium has its specific role and can also perform what it needs. So that's the forming of the team. Then you want, of course, to make sure that you have a high performance team under all conditions, you could say. So you want to make sure that combination of strains actually has all it takes and all it needs to perform also in the patient's gut. So you're building functions, mechanisms that minimize the dependency on the patient's existing microbiota, and where you can make sure that the consortium, you could say, has its own support network also in the body to perform. And so when you have your high performance team with your building blocks, of course, you still need to be able to manufacture it and turn it into a drug. And there, Coral really differentiates on enabling the entire consortium, even up to those 30 strains or more, to be produced in a single standardized GMP-compliant fermentation process. Not just doing it separately, producing them all separately, but really integrating it in one single manufacturing room.
Yeah. So, I mean, let's stay on that manufacturing point because it strikes me as being genuinely significant. You know, producing a whole consortium as a single drug substance. How hard was that to crack, and why does it matter so much?
Today, we're not the only one working on those therapeutics based on combination of bacteria. But when looking back over the last 10, 15 years, we have seen that for many companies, manufacturing of those consortia therapies was indeed the major bottleneck, and even has crashed several companies because they just could not produce their product. And one of the reasons there is that historically, microbiome companies that had those multi-strain products, they decided, okay, we need to grow each of those strains individually in a separate process. So we need to then first develop individual processes, run them. Then we have individual strains which are produced, and now we need to blend them. But of course, it's easy to see that this is extremely complex. It's expensive because you have all these different processes. It's slow, it's difficult to scale. And basically, it also creates complexity at the regulatory front because instead of just one product, you actually have a combination of multiple individual products. Each strain, you could say, it's its own drug substance, with its own supply chain, quality parameters, stability data, etc. So we saw that from the very beginning, which was actually when we started already in 2014, so way before we incorporated MRM Health. We saw that a bottleneck and we decided, okay, we need to take the time to do it differently and come up with something that is effectively allowing those products to be produced also at commercial scale. And so instead of jumping immediately, we actually used one of my previous companies, Digest, as an incubator, you could say, to pass the seed stage of MRM Health and to focus really on the technology development, to set up an initial pipeline and make sure that when we would come out, we had cracked that manufacturing code and we had something that could rapidly progress through the clinic. And so it took actually 5 years to develop that technology. And only in 2020, when we felt ready to proceed towards the clinic, we incorporated MRM Health as a spin-out from colitis. And at that moment, we had a product that could get in, and it was very elegant to be produced. Now, what does it mean? The major breakthrough and the difference versus those other companies is that we didn't start producing all those strains individually. No, we made sure that we could bring all the strains together in one single bioreactor, in one single fermenter, and with that we could grow them all together in balance in a way that we could maintain the ratios, that we could maintain the viability of each of the strains. And so as a result, instead of having all those individual drug substances, we just have one drug substance because we have one manufacturing process, one biomass that we try into one powder, and that we can then also treat completely as a single active substance.
Talking about products, particularly MH002, which I understand is a sort of a product that involves 6 different strains. How did you actually select the 6 that were going to be part of the product, and what are they actually doing inside the gut?
Maybe to start off, this product was developed for IBD, and IBD in essence is a disease of the gut microbiome. It's a disease in which continued disturbance and dysbiosis in your gut microbiome leads to a mismatch between the microbiome and the host immune system. That leads to impaired barrier integrity, to continuous overstimulation of the immune system, etc. So, because it's a really microbiome-driven disease, we started also from the patient's microbiome, and we used the Coral's bioinformatics engine to analyze what is now the functional difference between the microbiome patient and of a healthy individual. And that could allow us to see which strains are consistently depleted, which ones could mechanistically be linked to the pathways driving the inflammation, and could allow us to select specific bacteria that may have therapeutic potential in the disease. So that's the, you could say, patient first angle. On the other hand, we also used kind of a bottom-up approach in which we already had a large library of isolated strains, which we could then screen for having specific activity against the mechanisms of the disease that we wanted to tackle. So kind of a top-down and a bottom-up approach combined. And ultimately, those two strategies, they led to that set of carefully chosen strains in which each strain was in there for a specific function. Some of those strains, they restore key functional processes within the microbiome, such as short-chain fatty acid production. And I think about butyrate, which is the primary fuel for colonicides and is very strongly anti-inflammatory. So one part was about microbiome, but also we selected strains that directly have an impact on epithelial barrier integrity so that it could break down the barrier and reduce the leakage of negative microbial components that continuously would overstimulate the immune system. So that's the second thing about the barrier. And then the third aspect we wanted to build in is strains which can directly modulate the immune system, not suppress, but modulate it, in which we tune the immune system away from the pro-inflammatory pathways and we push it towards the immune protective pathways. So it's really a very specific combination that allows us to tackle those key mechanisms. And of course, we used again Coral to optimize that team. As I said before, it's all about team building and creating high-performance teams, with as a result that you have a consortium in which the whole is greater than the sum of the parts. And so it's really about team building at micro level, which is also reflected, by the way, in the name of the company. MRM stands for Microbial Resource Management, which is basically the microbial alternative of human resource management, because also in the gut, the microbiome, it's about having the right bacteria, but also to make sure that they work together and function as a team. But having said all that, so the specific combination of strains in MH002 allowed us to create a product that is really fully differentiated from anything else out there, because instead of acting like typical drugs in IBD on one specific molecular target, typically leading to immune suppression, we actually have a product that combines a set of mechanisms that address true hallmarks of IBD: disturbance microbiome, barrier integrity loss, and excessive inflammation. And by combining that, it could really be a first-in-class product that can even tackle the root of the disease beyond only symptomatic treatment, And of course, it's validated in clinical trials. It could even be disease modifying and have sustained positive impact on the patient.
Yeah, I mean, disease modifying is clearly the holy grail for a lot of drug developers. I mean, the phase 2A results in, UC and pouchitis, I mean, they were genuinely interesting. Sort of the kind of data that actually makes you sit up. What, as a company, did you see, and what do you think the results mean scientifically and clinically?
We indeed deliberately set up 2 initial phase 2A trials at the beginning, and both were relatively small trials. One was 45 patients, the other one was in pouchitis and growth indication was 14 patients. But both trials had the goal to, on the one hand, assess the safety, and on the other hand, to assess whether those mechanisms that we build into the product effectively could be replicated and could be seen in the patient. So that was really the goal, to see if how we built the product also resulted in the efficacy signal that we hoped that would be shown. In the UC trial, which was a placebo-controlled double-blind randomized trial over an 8-week treatment period, we first of all saw indeed these products are, you could say, super safe. We did not see any treatment-associated adverse events. There were no adverse reactions to the product. It's basically a clean sheet on the safety front. But also on the mechanism of action, we saw that all parameters from the endoscopy to the patient-reported outcomes, etc., all moved in the right direction. So we did see that recovery of the gut microbiome with the shift from dysbiosis back to the healthier composition. We saw the mucosal healing at the endoscopy. We saw the anti-inflammatory effects at biomarkers at tissue level, but also when we did a more in-depth analysis of biopsies, so tissues that we collected from the gut on which we then looked at gene expression, we could really see that replication of the mechanisms linking to mucosal healing and anti-inflammation. And then, of course, also important, as a whole, we saw that there was this initial signs of clinical remission, which means that patients tended to completely recover within that period, and that we saw a clinically meaningful improvement of the patient's symptoms. So, main lesson learned is that indeed could validate that multifactorial mode of action, and that we, of course, saw that very strong safety profile, which is, I would say, a key differentiator from virtually any of the other products out there, like biologic, but also other advanced treatments, because those products with immune suppressive activities, each of them will have a certain risk, for instance, linked to infections, linked to malignancies, etc. And then also, and I'll be short on that, on the pouchitis trial, that was an open label study that basically we did see the same safety profile. We could see again the replication of the mode of action. And in that study, even half of the patients within 8 weeks went into remission. So we saw that we had a very powerful activity signal, small study, but at least a very promising effect. And that consistency between the 2 studies for us is really a promising de-risker and a promising step forward for this product.
So, and I mean, earlier this year, you secured FDA IND clearance for Starfish UC. What does that trial look like, and what are you trying to prove, and what would success actually look like for you?
Yeah, I can even go a step further, because we are now effectively screening patients in the Starfish UC. And just now, we also received confirmation from FDA that we have fast track designation approved for MHO2. So actually, we have very strong regulatory support also. Now, Starfish UC, it's basically a large version of the first trial. So it's a randomized, double-blind, placebo-controlled phase 2B trial, in which the main goal is to confirm in a very robust way the efficacy profile of MHO2 across Europe and US. And it will enroll about 204 patients. Again, mild to moderate UC like in the first trial. And it's intended as a second-line treatment in those patients. So it's for patients who are inadequately controlled with vitamin A. Maybe they also already take steroids, but they still remain sick. And at that moment, MHO2 can really provide a solution, because the perception today is that there's already a lot of medicines out there for UC, which in a way is true. However, it's mostly for the moderate to severe fraction, less for the mild to moderate patients. And even then we see that still a large fraction of the patients are not fully responsive to the treatment. And so what we there want to do is create like an alternative step-up medication for mild to moderate patients. So when they don't respond to the conventional treatments, typically today they are, you could say, forced to switch to the immunosuppressants and the anti-ulcer treatment. And actually what we want to do is give them an alternative, safe treatment modality that can help them in a safer way and also can, in a sustained efficacy signal, help them over the long term. So that's how we position POE. Now, the trial itself, it's a 12-week trial, remission induction again, so seeing how the disease is going away, and then followed by a 40-week open label extension. And of course, what do we expect or hope to obtain? We hope that confirmation of the efficacy signals at the level of the endoscopy, clinical remission, microbiome, etc. And this with that clean safety profile as before. And the fact that we could get that IND clearance, the fact that we now also have a fast track designation for MHO2, that's really testament of the regulatory support and the potential, you could say, of MHO2 to transform the lives of patients living with UC. And Starfish will help us to further validate that and bring that convenient immune-sparing one-pill solution closer to the patients. And so data readout will be around Q4 2027.
So beyond MH002, you're working on programs in other inflammatory diseases and also in immuno-oncology. Now, the IO piece is particularly intriguing. So what's the thinking there?
It began based on quite recent science that there is now a quite compelling evidence that the gut microbiome significantly influences not only how the immune system functions, but also how that then in turn impacts how patients respond to immune therapy, like, for instance, immune checkpoint inhibitors, which you could say are the drugs that have transformed cancer care. So what has become clear is that in order to have that response to immunotherapy via the immune system, you actually need to have a healthy, diverse microbiome. If that's the case, we see that there is quite a high response to immune therapy. But if it's not the case, if those patients have dysbiosis, they often just don't respond at all. And there's the basis also of the problem. Many of those cancer patients, they already had a history of either antibiotics intake or they had previous radiation or chemotherapy, which are all factors that negatively impact your gut microbiome. And so together with our academic partners like Emil Woest from the Netherlands Cancer Institute, what we want to do is to develop a novel microbial therapy that specifically modulate, you could say, that gut immune axis in a way that it restores microbiome and that via that restoration, it can enhance the efficacy of checkpoint inhibitors. So what we don't want to do is replace the immune therapy, but actually we see that via the microbiome, we can make it work better, more effectively for patients. And of course, if even a fraction of the non-responders would become converted to responders, it had an impact on oncology in general, which would be enormous. So that's one of the two programs that we're now pushing towards preclinical proof of concept. And then in parallel, we have a second program targeting against inflammation, not only within, but also beyond IBD. And while in this case, Crohn's disease is the first target of the program, we actually develop it as kind of a pipeline in the product program where we see opportunities for that one product across a range of different inflammatory diseases. And that's maybe a core message about Coral. It's not a single technology that applies for a single disease. No, it's really disease agnostic in its design capabilities and works, you could almost say, wherever dysbiosis of the microbiome is mechanistically linked to the disease.
Yeah. So let me try and pull this together, what we've heard today, because the story here, there is a beautiful arc to it. I mean, we started with the science. The gut microbiome has a critical regulator of health and disease, but there's a frustrating gap between knowing that and being able to do something about it therapeutically. The fecal transplants, too crude, single strains, too simple, and previously no one had actually cracked the problem of designing, optimizing, manufacturing a defined consortium of bacteria as an actual drug. Before I let you go, Sam, what is the single milestone that would tell you that MRM Health is truly delivering on its founding vision?
I think that's an overswell. The day that the first IBD patient walks into the pharmacy, picks up his or her prescription for MH02 as a single daily capsule, and can begin that treatment with a product that truly restores the gut microbiome, heals the mucosa, controls the disease, and this without stressing their immune system. So it's really about, I would say, the application of our vision. We believe we can really redefine how immune-mediated diseases are treated by harnessing that microbiome to restore immune balance.
Sam Possemiers, founder and CEO of MRM Health, thank you for what's been a genuinely fascinating conversation.
Thank you. And again, thank you for having me on the podcast today.
Yeah, so for listeners who want to learn more, head to mrmhealth.com and take a moment to appreciate the journey that this company has made from a university spin out in Ghent through a pandemic era launch to a global clinical trial backed by an international investor syndicate. Here on BioBiz Buzz, we cover the full breadth of life sciences innovation. But every once in a while, a story comes along that actually makes you rethink what a medicine can be. The idea that a carefully designed team of bacteria, species that already live inside us, can be developed, manufactured, and prescribed to treat chronic disease is clearly a story worth following. I'm Mike Ward. Thanks for listening. And don't forget to subscribe to BioBiz Buzz on your favorite podcast platform to keep updated about future episodes. Until then, it's goodbye from me and keep on innovating.